Researchers from the University of Illinois' Urbana Champaign have developed an ultrasensitive test using a paper-based electrochemical sensor that can detect COVID-19 in less than 5 minutes. This research, led by Professor Dipanjan Pan, is published in the journal ACS Nano. As of today, there are only two main test categories. The first one is by using reverse transcriptase real-time polymerase chain reactions, or RT-PCR, and the second one is by using nucleic acid hybridization strategies to identify the existence of the virus RNA. On today's video, we have listed some researchers' development in health that maybe can detect the existence of COVID-19 virus in someone's body. The development of point-of-care biosensors. Researchers recently announced their successful inventions in creating point-of-care biosensors using 2 dimensions nanomaterials such as graphene to detect disease. The main advantages of graphene-based biosensors are their sensitivity, low production cost, and fast detection turnover. The research team created a graphene-based electrochemical biosensors with adjustable electrical readings to selectively detect the presence of SARS-CoV-2 genetic material. There are 2 components to these biosensors: a platform for measuring electrical readings and a probe for detecting the presence of the virus RNA. To create these point-of-care biosensors, researchers first coat filter paper with a layer of graphene nanoplatelets to create the conductive film. Then they place a predefined design called electrode on top of the graphene as contact pads for the electrical readings. Both gold and graphene have high sensitivity and conductivity, which make them very sensitive to detect changes in electrical signals. On current RNA-based COVID-19 tests, the RNA is used to filter the presence of the antigen in the SARS-CoV-2 virus. In this study, the team designed oligonucleotide antisense probes to target 2 antigen regions. This step ensures the reliability of the sensor in case there is a gene mutation. The gold nanoparticles are further coated with a single-strand nucleic acid, which represents an ultrasensitive sensing probe for SARS-CoV-2 RNA. Test sensor performance. The research team tested for the performance of the sensor using positive and negative samples of COVID-19. The sensor shows a significant increase in the positive sample voltage compared to the negative sample. In addition, the sensor also confirms the presence of a genetic virus in someone's body in less than 5 minutes. This test is widely used in many countries as it is known for its portability and low-cost production. The sensor on this test can also be integrated with microcontroller, an LED screen, or with a smartphone via Bluetooth or Wi-Fi, so you can also use it on any places. The research team also estimates that this test system mentioned above can also be adapted to detect various diseases.